And then 3 is how can we click continue? There we go. Is how do we characterize that, or what's up on the horizon for the stuff that we're doing in our research lab, and many people are doing in different research labs across the U.S. and globally. So let's go ahead and get into it. And I also want to point out that although my name is here, I was too lazy to write everybody else's name for collaborations, but there's many other graduate students and research staff scientists and professors working on this over the duration of the past couple of years in this, and really strong collaborations with the University of Wisconsin. So we're keeping the Midwest theme alive in this presentation as well. Let's see if we can get clicky click to work. All right, so a quick primer on radiation effects and materials. And so the primary motivation of this is really about power generation. So you have many different ways to generate power. One of those is the nuclear reactor, where you're using either fusion or fission in order to generate heat. You turn that heat into power, right? It's a pretty common type of method. The problem with that type of power generation is ultimately going to be you involve neutrons, either in fission or fusion. You're going to have these high energy neutrons moving around, and they're going to create damage. You can think of it to a certain extent of somebody sitting there with a gun and shooting bullets at a wall. You're going to create these types of defects or problems with your material. And so as somebody that would be operating a reactor, I want to know how that changes my material's performance, right? So what is my susceptibility to degradation due to radiation? But in reality, in most applications, particularly for structural materials, so that means things that are going to hold your reactor together, radiation isn't the only factor. You also have to consider that you're going to be applying some type of stress. You may be having some type of corrosive media. And the simplest reactors that we run in the United States right now, that would be water. But on the right, we show many other types of reactors that are being considered. So generation 2, generation 3 is our current reactors that are operating. And all of these ones that are proposed, you may have seen a lot of news lately about Bill Gates and his company or his venture in TerraPower. That's over here. There's a lot of energy in startups to try to, pun intended, generate more energy through these different reactor types, where they're going to go to much higher temperatures and much higher degradation levels in the material. So we've been operating Gen 2, Gen 3 light water reactors for a long time, commercially since the 50s and 60s. And what we wanted, so we understand this quite well, we need to migrate towards this particular area here. And so a lot of the research based around this is understanding how this radiation causes that degradation, and then going into more complexities as you increase the different types of what we would call stressors, right? So stress, water in there, and changing your material, of course, also changes its response to these different factors as well. And so that's kind of the basis for the talk and a lot of the research that we're doing with the in-situ TM facility that we have here on campus at University of Michigan. So what does this look like in real life? So these are just some conical examples of what we would call radiation effects, right? So you have this essentially mesoscale type of response, where on the left here, this is a piece of metal, right? This was never put in a reactor on the left, and then they put it in the reactor, they take it out, and this is what happened on the right, which is, we call that swelling. So the material has basically changed its volume in the reactor. You can imagine if you're trying to run something where you're running cooling up and down in your particular reactor, this swelling response would close those cooling channels, and you would prevent cooling and have meltdown. The number one thing in our department is you don't melt the core. So that would be a very bad type of thing, right? So this is a conical example. Here's another one. These are fuel elements in a light water reactor. This is where you would have water coming up these fuel rods up here. Of course, you probably wouldn't put them in wavy like that. They're supposed to be straight, like on the left here, but you're actually getting a radiation response called radiation growth, where it's actually moving and causing this fuel element to bow, which is also would be a bad day for reactor operation in it. And then here on the right is an example. This is a bolt that would have been holding down what's called a baffle plate. that sits outside these fuel elements, where the radiation has effectively totally embrittled that material and made it so when you have a stress component applied, you get this cracking. It's called radiation-assisted stress corrosion cracking along this baffle bolt. You would imagine a cracked bolt means that it can't carry load anymore, so it's not doing its functional component as well. And so these are all kind of common, or I wouldn't say common, right? These are possible degradation modes. These aren't happening every day in a reactor, but they possibly could. And so we need to understand how different materials will perform under those different responses, and if they would have some type of degradation mode, such as these kind of 3 examples shown here. And so if we take that down to the radiation effects to its most fundamental level, right, that I'm showing essentially the engineering scale application, but if we look at it at the simple physics, it's quite simple. Most structural materials are going to be crystalline. So that means that they're going to have their atoms in periodic arrays in the material, right, or crystal lattice, right? And you're going to have an incident particle with energy. So in the light water reactor examples I showed before, that would be a neutron, but in reality, that could be an ion, that could be an electron, basically anything with a mass and given energy associated with it. If that particle comes in and impinges across that material, it can interact with those lattice atoms. And so if it has enough momentum or effectively enough energy and it can transfer that to the lattice atom, you can actually displace that lattice atom from its functional site on the material, and that incident particle will continue to go on if it has sufficient energy after that energy transfer. This will continue to happen where that first atom that was knocked off will then interact with other atoms. You'll continue to have more interactions. We call that a damage cascade. And eventually what you'll end up with is a bunch of defects that occur in your material. So say in this site here, or this original site here or here, we now have vacancies, right? So we have our lattice atoms displaced from that site, which would be a vacant site or a vacancy. And then we also have our original incident particle and then other lattice atoms that were displaced that have come to rest in positions that they shouldn't, which is an interstitial. And so we generate these Frankel defect pairs. So that's kind of the simplest approach to radiation effects. That's why I like the analogy of shooting bullets. You're essentially shooting bullets here and making holes and shrapnel in your material. We're trying to understand what's happening with that type of response. And if you give enough time to these types of defects that produce and enough energy, of course, they will migrate through your material through diffusional processes. They can agglomerate together and they can form extended defects. And it's those types of extended defects that we observe primarily in the TEM, using transmission electron microscopy. That's a large part of the what my research group does, is using TEM to look at those extended defects in the process. And so if you look at it at a little bit longer length scale, so I just talked about point defect creation here through that kind of simple bar napkin type of drawing before, and when you can have those vacancies interstitials, they can cluster, they can recombine, right? If you have a vacancy and interstitial combined, then it goes back to a normal lattice, or they can be annihilated, which means that they go to a free surface or a grain boundary where it can actually absorb that defect and not have major effect. But what we're primarily interested here is in clustering, where we get these voids and dislocation loops. And I'm going to focus on these dislocation loops, which essentially just means that you have a line dislocation that completely encloses itself within the material, making a loop, which means that you can use conventional strain-based contrast imaging or G.B imaging in a TEM or in a STEM in order to characterize those types of defects, right? And if you wanted to, you could take at Michigan and many of the universities you just said, there's classes talking about this radiation effects if you're interested in it in more detail, but I think that's enough to get you kind of talking generally about it. But if you think about that, the creation of point defects, the creation of extended defects, those all will dictate the much larger engineering scale properties. And so that comes to the point that this is an inherently heterogeneous and multi-scale problem, right? So you're going all the way from the simple atoms all the way up to a large engineering structure. And so the question becomes is how do we do these observations of the radiation damage? How do we quantify it? How do we characterize it? Do this type of responses? Yeah, so apparently that's already there. And I already alluded to it that we're going to talk a lot about TEM and STEM, primarily because we're interested in talking about in-situ TEM based techniques. But there's the full gamut all the way from optical SEM based techniques and atom probe. And there's a bunch of complementary techniques such as small angle neutron scattering, which is really interesting type of technique. If you ever get a chance and you're not familiar with it, it's a fun one to do that correlates well with APT and STEM, quite nice. But we're going to focus here primarily on STEM. I'm showing both the EDS spectra on the bottom and a diffraction contrast image on the top. We're going to stick with diffraction contrast imaging for the day, right? So we're going to be talking about these types of guys here, which is a TEM column. I figured it's primarily for a student chapter. So this was me in my first year as a student doing microscopy at Oak Ridge National Laboratory. That's a Philips CM200, a precursor to the FEI Tecnais that are now available in it. So you can see the control interface is all physical buttons instead of computer clicking and stuff like that. I really love this system. This is one I actually wrote my thesis on effectively in there, but we've progressed quite a bit from here. But the principles that worked then also work now in today's environment. Okay, so we understand at least a little bit that radiation effects is primarily considering the production of defects, and we're going to use the TEM to characterize those, right? But then the question becomes is, how do you simulate these effects, particularly at a university, right? So in general, you aren't going to do neutron radiations at a university because you're going to activate your samples. They're going to become particularly hazardous. It's a difficult process. And those irradiations can take on the orders of years, if not decades, to do. So many students will want to graduate in 5 years or less. Right? So if I told them they had to wait 10 years for neutron radiation, they would never join my group. So how do we make them join the group, and how do we simulate this? We use ion beams, right? So instead of using a neutron, we can use a proton, or we can use something like a heavy ion that you generate out of a source. This is a schematic of the Michigan Ion Beam Lab. We have a bunch of different sources on 3 different beam lines here. So you can generate anything from a proton all the way up, I don't know what the high Z. It's a ridiculous number of elements on the periodic table, both solid cathodes, gas cathodes, the whole gamut in there. You would generate those. You would then use an accelerator to get those up to energies that are relevant to create damage. And then you would bring it down a beam line and hit the material and create that damage process that I talked about, right? Creating those point defects and doing a damage cascade. The reason we like these is that they're extremely well controlled, right? So you can control things very easily, such as temperature, dose, and dose rate, where dose is a quantification of the flux or the fluence on a sample. In there, we can accelerate those irradiations compared to a neutron radiation as small as a factor of 2 and as high as a factor of 1,000 or more. In there, that means that you will get your thesis written in 5 years or less. And then, yeah, in a typical type of reactor, so TerraPower's reactor wants to do things where they're going to be about 100 to 200 dPa. So we can simulate that damage process in about 2 days for the TerraPower reactor. And then for me as a professor, the beauty is that they're cheap. They're very low cost, so I can do a lot of different experiments in this process. And so again, this is the Michigan Ion Beam. So we would have sources, an accelerator, you do some optics. It's basically actually, when you think about a TEM, it's basically the same thing as a TEM, just laying flat, right? You have a source, you have an acceleration, you have a condenser lens, you have scan coils, all of those same things exist on a beam line. And the thing that I wanted to talk about, or was asked to talk about, is this guy sitting out over here, which is actually a FEI Tecnai system where we can accelerate 2 different ions at the same time, bring that coaxially into the ion beam, and then irradiate our specimen and see that damage occur dynamically or in real time, or the fancy word, of course, would then be in situ to do that. And so this is the facility that we'll spend a little bit of time. Before we do that, we should probably do a little bit of a history lesson. So when was the first time in-situ TEM ion radiations completed? The first paper is by these 2 right here in 1961 with the name of Ion Damage to Metal Films Inside Electron Microscope. They used a Siemens system shown here on the right. And what they ended up doing is observing small spots and loop features form intrinsically in the material as they were looking at it, right? So they were seeing this defect formation, the extended defect formation occurring in a TEM in that process. And so they identified those as possible dislocation loops and stacking fault tetrahedra, which we now know are very common to form under irradiation. Back then, it wasn't necessarily the case. And they did a bunch of tests and had a bunch of hypotheses associated with that observation. And they basically came to the final conclusion that they are getting this radiation damage to occur due to the oxygen ions being emitted from the filament. Right, so this is a very old microscope, not ideally suited for today's type of microscopy efforts, but it used a tungsten filament with some oxide on it, and that was getting accelerated with the electrons, creating that irradiation damage. And so just like penicillin was kind of discovered by accident, so too was the possibility of doing in-situ microscopy by using the electron gun as also a oxygen ion gun that coaxially accelerates down through the sample, and did it that way. And of course, this is a previous slide show, and as I will allude to, that's not the way we do our in-situ TM experiments nowadays. And we now typically use an ion accelerator or some type of source in order to generate an ion. The most common ones that you would see nowadays are primarily gases, so xenon, krypton, argon, those types of ones, because they tend to be very stable sources. You would then accelerate this down a beamline. You have some defining apertures, and then you also have essentially condenser and bending lenses that would occur. And then you bring that in through the beamline, you bend it down using electrostatics, bring it in, and then your TM sample would be sitting here, and you would have your electron coming down at the same time, and you would view that observation of radiation damage down there. So this is a old picture from 1974. So remember, the first irradiation was done in 1961, or first observation was done in 1961. They then very quickly progressed. So there was a huge boom in the late 60s and early 70s of in-situ radiation facilities. They actually published all their designs. So pretty cool to go look at the pictures of these here. But this is then 1974. What does it look like if you move forward a couple decades? It would look something kind of like this. So this is the IVEM facility at Argonne National Laboratory. So this is the intermediate voltage electron microscope facility where they have Hitachi TEM system, and then they have their ion beam coming down through the sample. And so what they've actually do is they mill out a portion of the pole piece of the microscope in order to bring that ion beam down the column. They do a last minute bend, and then they're coming right through that pole piece. You can see that cut right here in order to get that ion beam come down and hit your specimen. And then your electron beam, of course, would stay the same. You would imagine that degrades your resolution and your optics. It does a little bit, but not significantly enough, because we're very infrequently doing high resolution type of microscopy, right? We're doing typical strain-based contrast, things that you learn that you can image dislocations, but rarely actually do unless you're doing stuff on structural materials and that type of process. But this configuration is quite common. And so this is the IVEM facility. This was probably considered the most premier facility for doing this irradiations over the past 2 or 3 decades. I think they commissioned in '95, if I remember right, in there. But in reality, you can actually do a bunch of different types of configurations for ion beams. So here's some just examples. This would be your upper pole piece. This is your lower pole piece. The green part here is your electron column. The red part here is your ion. Black part's your sample. So you can either come in through the side, so you don't have to do any crazy things with your pole piece. The problem becomes is the angles that you have to tilt to. Remember, we're doing diffraction-based contrast, so we need to tilt our crystals correctly, right? And so that becomes a complicated experiment, which is why many people will come in and mill out the pole piece to try to lower that angle so that it's much easier to try to get everything orientated correctly. You can also use electrostatic deflection to come in over the top of the pole piece and come down. And then now with changes in pull piece designs that can actually be much larger, you can actually potentially bring in, say, 2, 3, or 4 different types of beams using a mix, say, of the 2 different types of techniques up here in that process. And so the facility that I'll talk about is using primarily this drilled pull piece type of device in order to do that. I'll pause there real quick. Any questions up to this point? Probably going way too fast. No?
I think the speed is good.
Yeah. Okay, thumbs up. Sounds good. Okay, so, you know, about 5-10 years ago, University of Michigan, we had all of those accelerators and everything else that existed and went through a major upgrade. Actually, I have the coaster for when it was recommissioned, 2015. Okay, so 6 years ago. We recommissioned the lab, but as part of that, we wanted to build this in-situ facility. And you can imagine the huge amount of advantage of doing your observations in situ, because you can look at the same region of interest and see those defects grow and move, and you can really tie that in quite nicely to modeling types of techniques. And so we wanted to have that facility, and so we developed through a survey. I think there's about there's over 10, less than 30 in-situ irradiation facilities across the world. Did a survey and realized that we could do it. So what we did is we bought a FEI Tecnai, which now be a Thermo Fisher Tecnai column. What nice thing about this is that it has a large pull piece, which means that you can come in through the side here with your ion beam system, right? So this is a fancy flyer from when we commissioned this device, but this would be a Tecnai. If you realize and you know your generations of FEI, Thermo Fisher, and Philips systems, this is basically that same CM200 column, just dressed up to look a little bit fancier with digital controls on the right set of hard buttons. And then you have the ion beam coming in through here. And there's a whole bunch of science and engineering involved with trying to get those beams to come in right, having them to be the right volume, considering the different angles, and then also considering that just past this part over here, you have a bunch of turbo pumps and cryo pumps hanging off the ion beam, which causes vibrations. And so you have to consider vibration dampening in there. So there's a lot of complexities to commissioning a particular type of machine like this. Luckily, I wasn't here when they did that. So my predecessor, Professor Gary Was, directed and developed that components in there. So I got to skip the hard part and I just get to use it now, which is the awesome place to be in. But this is also then our configuration. So if you look down through, like if you're an electron basically going down, you would have your ion beam coming in from the left. Your CompuStage stage 2 goniometer would be down here. And then we actually have a viewport where you can see things come in from the side here. That's this guy right here. And you would have these types of angles when you're trying to set up these particular types of experiments while also trying to stay in the correct diffraction vector. You also then have to think quite a bit about all the different angles involved. Because if you take a side cut, this would be your sample, your electron beam's coming down. Your ion beam is 68 degrees off axis from the electron beam. So you have to consider all of these different tilting things. The one thing that's particularly unique or nice, I think, about our system is that it's a little bit more modern. And so we have a 1K by 1K CCD detector running at 30 frames per second. San Diego National Laboratories is probably the only one that outpaces us in the US for capture rate. And we have a MEMS heating holder. This is a dense wildfire TEM stage here, where you have these 4-point probes that you would put a MEMS chip on and then use that to heat your specimen. These can go to much higher temperatures at much faster ramp rates, so you can do crazy things with ramping up and down or oscillating temperatures and all of those different functions there. Okay. So this is what you would do if you're going to load that on the MEMS device. So this is a dense wildfire chip here. You have your 4 contact pads right here. Inside you have your heating membrane that's pictured effectively right here. You would fib out a specimen, so use focused ion beam in order to make a nice little fib lamella. You have to consider all the crazy angles that you're going to have. Land that onto the chip. You have all these different holes in where you could place them. You would then thin that down, weld it, put it on your specimen, and then that makes your specimen for doing the ion irradiations part of that. The nice thing about doing a fib-based technique compared to the previous electropolishing techniques is that you can now do targeted lift outs. So you can say, I want a 100 zone axis or a 111 zone axis, whatever crystal orientation. You can lift that out, land it, know you're going to be there. So if you do the fancy things with considering angles, remember your geometry from high school, and then also know your crystal orientations, you can sort out all of those details in order to get a perfect specimen. The only problem with this type of technique is that you have a limited field of view, right? You're only looking at a couple microns in your field of view, so you don't get very good statistics. But we think that we'll have good advantages across this. So our system has only been truly operational for about 6 months. In my research group, we just had our first successful irradiation just a couple weeks ago. And so I'll show that here. So what we're doing now here is imaging in dark field mode. We're looking at so that means that black is your background, or basically your perfect crystal, perfect in quotes. And then the white dots are your defects that are being formed under irradiation, because we have that strain-based contrast that's coming. And if you look closely and Zoom is broadcasting correctly, you can see white spots kind of come in out of contrast. Some of them join and come together. You'll realize that this is, and you just see a focus event occur there where we're just focusing the beam. You very quickly realize the complexities associated with doing these types of experiments because you have the ion beam, electron beam, both have to be focused, both have to be vibration minimized or stabilized. So there's some camera stabilization going in here. And it's a difficult experiment to actually run, and we're still trying to currently optimize those to get it correct. But this at least gives you kind of an example of the type of images that you can get. I'll show much better ones in the future. But you can kind of sit here and squint, and you can see defects being formed in the material. All right, so let's move on. So if you take the screen grabs out of it, these are the types of frame-by-frame captures that you would get. So we're going from a pristine material. All these white guys here are actually precipitates that are formed, and then you're going through a damage event. So we use this factor called displacements per atom. We don't have to get into the detail, but it's just a quantification of damage. Typical reactor, typical light water reactor would have a DPA of about 16 for the fuel elements in it. TerraPower wants to run their structural materials to 200. So this is just a fraction of that. But what you're seeing here is that these, we believe the precipitates are coarsening, and then we're also getting the defect or dislocation loop production. And so what my graduate students trying to do is ultimately quantify the coarsening events versus of the precipitates versus the loop formation, and compare that between different cluster densities, right? So you can imagine that if you increase the number of precipitates, that you would increase the amount of sites that those defects could go to, which would limit radiation damage to occur in the material. So this is kind of a first result, not completely optimized process, since our facility is still kind of in its, still, I would call it an infant, quickly moving to being a toddler. And we'll get to its youth adult stages hopefully by next year at this point, but we still have a lot of maturity to go. Yeah, so what alloy and G-factor is being used here is a good question. So this is actually a really cool material. So we call it an additive nanostructured alloy, or ANA for short. What it is, it's a 9-chrome ferric martensitic steel that is manufactured using additive manufacturing. And by using AM, you can intrinsically trap more elements than you normally would by conventional solidification, which means that we can push our precipitation into crazy high number densities. So we have like on the order of 10 to 24, 10 to the 25 of number density of precipitates in the material in it. And I don't remember the exact G factor that my student was running, so I can't answer that, but I could figure it out and email it to you. In there, but we're using that in order to do that. Typically for BCC steels, biocenter cubic steels, you'd be imaging on the 100 zone axis, imaging the, I don't remember all the G vectors, 111, 011, those types of G vectors in there. There's some nice papers that describe that in quite grand detail. Good question though. All right, so you're probably thinking that's crappy pictures. Why am I talking or watching this when the microscopy is so bad? And again, I blame it on the fact that we're still learning on how to operate our in-situ microscope. But argon, which I talked about previously, IVEM, has been doing this for a long time. And so this is an example from my research group from a couple of years ago where we actually did the irradiations at argon. We irradiated it at high temperature, so 320 degrees C, which happens to be right around where its typical reactor operates. Again, here we're using gaseous ions, krypton ions. And what we're doing here is looking at 4 different alloys. These are called iron chrome aluminum alloys. They're bicenter cubic materials. They're being considered as replacement materials to prevent things like Fukushima from ever happening again. But we wanted to get an idea of what the composition content, how these defects would form as you change the composition, which is why we have these 4 different alloys. And oh yeah, this one's good. So we use the G011 for this one, imaging off the 100 zone axis. You get 2 different types of loops that form in this material, a 111 and a 100 loop. So the 100 zone axis, if you do all your G.B criteria, tends to be the perfect one to image both of those loop types associated with it. And then this here then is the just a GIF showing GIFs of each one showing the production of the defects and the growth. So if you look like right here in this image, you can see this loop growing and it eventually gets to the actual loop that's formed, and you can image that in the same region of interest. And so the nice thing is that you get very good growth rates as a function of dose across these different compositions, so we can ascertain the performance of the different materials. And you can see that across all 4 of these here. Sorry if I'm giving you a seizure by the amount of data that's kind of running at the same time, but it's kind of the easiest way to show it without running over for 2 hours, clicking on every movie. All right, so if we do that type of in situ irradiation, and this is just a cut form showing that loop growing that I had highlighted before. So you can see this actual loop growing. This is the G vector here in the scale bar. We're speeding this up 125 times. So a typical experiment can take anywhere from a couple minutes up to about half a day or full day in a process to do this. But you can clearly see that this loop here starts as a little black dot, grows to a loop, and then it continues to grow throughout that duration of the experiment. This trend is consistent across the 4 materials that we were looking at. So these are nice size distribution violin plots showing the dose of the 2 different loop types. It doesn't really matter to know what that means, just that there's 2 types. You can quantify them, and showing that you generally get your size distribution starting to tail towards larger sizes, even though potentially the average size or median size might stay roughly the same for the red loops here, or the 111 loops, and then your 100 loops do tend to grow as you continue across in it. And so through that in situ technique, this is done at the discrete points here, but you do get that dynamic observation where you see that this isn't growing because it's defect coalescence, it's growing because of diffusion and absorption of those point defects that are occurring in that process. And so we can then use these types of data in order to do effective modeling that might be able to predict towards larger doses or some other function in there. Okay, more data that you can get from it. Typically for material science, you'd be interested in size and number density. So I just showed you size. This would be the number density plots. So these again are your 2 different loop types as a function of a radiation dose, or essentially how long we radiated the material. And then the loop densities, and then the black eye here is the total density. The cool thing here is that you can just clearly see that, say, going from something that has about 10% chromium in its matrix, and just about doubling that, you suppress that density of defects that occurs significantly by a factor of about 2 in that. So you get, you can clearly then from these in situ radiations determine compositional effects as well as the dynamical effects in the process. And so that's the really, I think, cool power of the whole process here. That animation isn't there. So both of those, I think, hopefully give you an idea, or hopefully I've given you an idea of what radiation effects is, the different designs, the design of the one at Michigan, the fact that we're still learning on it, and a little bit of the data that you can get from it. But I think what you're realizing is that if you look at that previous slide, these doses, these data points, we didn't get those from looking at the video. We stopped the beam, did a bunch of tilting, got the images, and then counted them. Actually, it was about a year later, counted all the defects and put it in there. So you can start realizing quickly that with in situ irradiations, we have a data scale problem, right? The quantification that I showed you on the previous slide, I think we had about 1,800 frames total. We only imaged or looked at essentially 4 of them to look at the loops. So we're throwing away about 99% of our data whenever we're publishing our papers, right? So there's a very intrinsic data scale problem, right? Which comes to my point here that quantification of in situ dynamic data is currently hindered by human-based analysis techniques. So you can then start thinking about or coming up with ideas on how to improve that. But before we do that, we're kind of halfway through-ish, maybe a little bit past. So it's time to take a little break, play a little game, and understand why this is a particular problem. So I think you can already understand the scale problem, but there's also another one that we'll come to. So to demonstrate the scale problem and a couple other problems with our current ability for quantification of in situ data, we're going to play a little game called Count the Cats. Right, so this is Grumpy Cat sitting here. What I want to do is in the chat, if you can warm up your fingers, we're going to see how quick people can respond to answer the number of cats in image. So you don't have to do it in this one, right? We all know it's 1, but I'm going to flash the next slide, use the chat, put it in there, and we'll see how accurate people are, and then we'll see how long it takes people to respond. All right, so we'll start with the first one here. All right. So give people a sec here. All right, so if you look at the chat, everybody is coming in at the same number, right? And it took probably about an order of just a couple seconds for people to acknowledge it, probably another second to type it, right? So you guys are all correct, right? Our correct answer here is 4 cats. And we had some image complexity, but the background was pretty consistent. And we had just small morphological changes of our features here, right? Now, if we think about our in situ data, it's going to look much more like this, right? So let's see how long it's going to take for people to count the cats and what number they're going to get here, right? So we have a very complicated background. We also have features that we're not interested in, and we may have cropped features that we are interested in. So we'll give people a sec here to give a response. I think we captured the critical mass here, which is always a great pun when you're in any good department. So what we have here, then the correct answer is actually 19. Right? So nobody answered the ground truth, right? The real answer, because I think everybody has a particularly inherent bias in the way that they're going to respond to this answer, just based on what they know what a cat looks like, right? And so, and maybe you have some ambiguity also because you weren't sure if this was cat fur or dog fur or something else associated with it. And so this hopefully demonstrates a point that it took longer, so your scale is an issue, and we're starting to see an intrinsic bias. So the next question becomes, we start thinking about the videos that I've been showing previously. Now we can play the game here of count this, right? This is looking much more like the real images that we would see in the type of experiments that my research group is doing, right? And so I implore you not to be a scientist or an engineer, right? Don't count the columns and rows and then multiply to get the right answer, right? A true scientist would investigate every image at detail, make sure, decide that it's correct, and then count it, right? But I won't deplore you with it or make you do that, right? So my correct answer for this is I don't know how many. I actually haven't counted it. I had much better things to do, right? And so this little game hopefully shows you that when you start thinking about in situ data, you have an inherent frame by frame, like even in a single frame, there's a scale issue. But then as you multiply the number of frames or how long your experiment is, this quantification that I showed for that previous paper that we did is really laborious, right? And so as a graduate student, I wouldn't want to do that many times. As a faculty member, I would feel bad, but I would still make you count all of those, right, in a long time. But it's not an ideal situation. And also you would realize too, is that if I gave those videos to you guys to count, you all get different answers. And so how do you make a quantitative assessment across different research groups investigating similar materials or similar responses, which is so common today in collaborative environments, right? And so this becomes this problem with this 99% data throwout in it, right? We only looked at these 4 frames in order to generate that data, or 5 frames to generate that data. So one of the things that my group has been focusing on for the in-situ data extensively for the past year or two, and with the University of Wisconsin many years before that, was looking at automated defect detection, right? So I think almost every graduate student who does electron microscopy at detail has thought about making this really great Python program that does Huff transforms and finds those defects. They code on it for 3 months and they realize I could have spent those 3 months and just counted the 5 images I had and been done. And I just wasted 3 months in that process. That's what I did in my grad work, right? That was in 2012. That's when I finished here. So I was sitting in this regime. But in 2014, we saw this evolution to these deep learning based detection methods, right? So these convolutional neural networks, these are the things that are driving Teslas nowadays, right? Self-driving cars, your Face ID on an iPhone, if you have one of those and you're not an Android user, is using these types of detection methods. These have revolutionized the way we can do detection, classification of features in an image. And what we've been looking at and trying to apply is, does that work for microscopy? And does that work for our microscopy? And many other groups are looking at it for their microscopy as well. So I want to throw this in here because I think you would be interested. I'm only going to talk about the use of YOLO code, which means you only look once. It's an unfortunate acronym when you're trying to give a science-based talk at a conference, but we reuse the YOLO code here, but we also have data and recent papers out on the faster RCNN and master RCNN. These run a little bit slower, they're not as lightweight, so they're not as good for in-situ video. But I'll just talk about the YOLO stuff, but happy to talk about the others if people are interested. I'm not going to talk about the structure of convolutional neural networks in grand detail at all. The main thing you need to know in order to understand the rest of the things I'm going to talk about is that the way it works is you have a database of labeled images. Right? So these are STEM-based images doing dislocation imaging in there where I painstakingly labeled all these. Yes, it was me. This was before I was a professor and sat in an ivory tower and told other people what to do. We labeled these. You then put that into the deep learning network, right? So this is called the training phase. It will learn your labeling to a certain percentage. And then once it has reached a specific learning rate, you stop it, and then you can then input a new image, and then you'll get an auto label image off the back end. All right, so the main requirement for doing this type of technique is a little bit of knowing how to do Python, and then having a sufficient database in order to do the training. Right? And so we are finding that you can do this with as few as just a couple tens, if not hundreds of images, but your performance really, or what we call learning rate, really increases when you get to about 1,000 or more features of each individual feature that you're interested in. The other thing too here, this is faster RCNN results. You can actually detect multiple types of defects, which is why we have the different color coding here. Okay. So what we did is we took that old in-situ video, it's 5 years old now at this point, that was done at IVEM, and applied this YOLO module. So what we do is we take a video inside the Python code, we would split that into multiple frames. We use the YOLO module using convolutional neural networks in order to come out with bounding boxes, or essentially regions that it proposes would actually have a defect that we're interested in. So in this case, it's the black spots on the white background. And then we apply 2 modules after that. One is static property analysis, so getting things like size and number density, like we had shown in the previous hand labeling efforts. But now we're also then capable of doing defect tracking. So now we have dynamic property analysis module. And then your end output will be defect number, so defect 1, defect 2, defect 3, and so forth. You have a function of time or dose, and then you have all of this different data associated with, such as size and location, shape, many other factors that you could put in there, circularity, all these crazy things that you can data mine at that point. So this is the architecture we've been exploring. This is it working. So this is a GIF where we did the image detection. We combined those frames and make a video showing the bounding boxes. So this is only just a couple seconds of one of those videos that I was showing before, but you can see the blue guys here is your bounding box, black guys that are showing up in the image are the defect. And generally speaking, we're getting relatively good detection even over this dynamic process that's kind of occurring in the data. And if you compare the way we would have labeled it versus the way the YOLO would have labeled it, where YOLO is in yellow, humans would be in cyan here. These are the different bounding boxes. If you look at the overlap of those, we see generally really strong agreement. In the machine learning world, you would typically use what's called an F1 or F beta score, which an F beta is just a weighted factor of the F score. And if I send out, just in the same game of playing cats, we did this with images, sent it out to a bunch of microscopists. If I send images out to microscopists and ask them to label the data, they get an F1 score, which is basically a mean between the number of defects and the accuracy of that defects. They get an F1 score of about 80%, humans do, right? So they're getting about a B minus. And if they're really good, they might get about a B or a B plus, right? It's about 85%. The YOLO code, in the case of these particular data, it's kind of highly constrained for this particular experiment, but we were getting F1 scores of about 85 to 93%. So we're getting human level performance, if not above human performance in our detection. But now I don't have to have graduate students or other researchers do that labeling. I just let the machine run it. You can also think about that. I could give you the code, you could run it on the same images and get the same answer. So we're removing bias and increasing our temporal and spatial resolution in our analysis. Just to prove that it works, this is an example. So on the left here is the reference data. This is what we originally published, and a simpler chart than the violin plots that I was showing before. It's only for one of the alloys here, showing the defect growth as it continues to go up. There's an invisibility criteria that we have to worry about with GW vectors in just the video. So this is the ground truth labeling, taking that into consideration and the growth rate. And then this is the YOLO outputted size distribution on the right. And so all of these are showing the same trend, which is a monotonic increase with increasing damage, which is what you would expect. And generally speaking, the median or average sizes are roughly within the air of each other in that process. And so we're now getting human level performance with very little effort, to be honest, at this point. So this is really cool. What's even more cool, in my opinion, is that you can look at individual defects. So this is just one defect that we picked out of the image frames across the in-situ experiment. And then this is its growth chart, right? So just like you would measure your growth over the years as you were a little kid, same thing here. We have dose, defect size, and you see this very nice linear curve for this particular defect. If you pull out multiple defects, you'll find that some of them don't have a linear trend, right? Some of them will lay over much earlier or have some type of other functional form or shape associated with it. And now you could then take that data and say, oh, this one's linear because it only has small defects around, right? Or this one's parabolic because it has this response or something like that. And so we're being able to data mine much more extensively our in-situ experiments, which is going to give us much more insights about the material's performance under these in-situ irradiations. Last example, this is the last example or comparison between our previous paper and what we're doing nowadays. This is the previous paper publication on number density that I had showed, where we had showed that the 18 chrome is completely suppressed in the number density compared to the other alloys, right? Only 5 discrete points this is the full detection across every single image frame for all the in-situ radiations. You're seeing some variability at low dose here because it basically is detecting background. And so you could cut these out and with a little bit of human inspection. But you can see the same trend here, 18 chrome, lowest guy sitting here, and then you have your 10 chrome as having the highest density. This matches the same response, but now we can see much better what the shape of that curve is, understand what that is, and you can extract much more information. You see these little jumps here. This isn't a problem with the machine learning code. It's actually slight changes in field of view and stops in order to do this analysis that's shown here. All right, so in reality, if we knew that we were just going to do video detection, we would have never done this analysis, and you get just nice, beautiful, smooth curve the whole time. So this is a little bit from doing some dumpster diving for data in order to apply this technique. All right, so now if you can track or you can detect individual defects per frame, if you can track those defects across all the frames, that means now we can track trajectories. All right, so now we know where our defects are moving, how far they're moving. And so what this is here is a plot showing those trajectories, or essentially their position across all frames for the defects. This isn't particularly interesting for the material system that we irradiated because they don't move in reality, but you do see some particular defects moving more, right, by longer trajectories here than you do, say, individual defects sitting over here. And this is a blow up of this region over here. So that means now we're actually able to quantify or track the defect motion, which for our field is really important because understanding that diffusion rate gives you an idea of how that material is going to respond as you continue to increase the damage, right, in that process. So that's a really nice result. If you take that data and you do a simple diffusion kind of coefficient or mobility method, you can then see the mobility as a function of defect size across the 4 alloys. And remember, the number density of the 18 chrome data was much lower than our 10 chrome data, and you can see directly why. So our mobility of our defects is significantly lower in our 18 chrome system than it was in the other alloys. And you can see that the mobility increases or is higher for smaller defects, but as they get larger, it basically drops effectively towards 0. So we're getting both the compositional effects and size dynamic effects for defect mobility, or tracking that in the process. And so from my viewpoint, and what I'll end on here is in-situ microscopy, in-situ radiation effects. We find it a little bit interesting in it, but I think the next breakthrough that we'll see is not in how we do the experiments or the TEM resolution or anything like that. It's really in this idea of automated data analysis and quantification that I'm showing in the kind of the last part here. Last thing to point out, that first experiment, if you remember, is 1961. So we're celebrating almost 60 years. So it's older than many of us here probably at all for in-situ radiations, which I thought was kind of interesting. But all these new breakthroughs, I think, are going to make the amount of science that we'll gain in the next 5 years will probably completely outweigh what was understood in the past 60, just by using that kind of automated defect analysis. So I'll leave that there with questions. Happy to have any discussions. I just highlight this last slide on the questions. So we are now actively working at University of Wisconsin with a startup company. Full disclosure, it's my startup company, in order to do augmented reality microscopy, where we actually run that defect detection in real time. So this is results that we just did yesterday, so 24 hours old, where we actually did that detection model in real time using a web-based app. It's pretty cool, and we'll see that probably online at MIBL, our radiation facility, in the next 6 months, hopefully. So with that, questions, discussion, happy to stick around.
Thanks, Kevin. That was a very interesting talk. I will, I guess, open the floor for questions. If probably you can just go ahead and unmute yourself and jump in if you have a question.
Any takers?
I have some questions if no one else does. So one thing I wanted to, I guess a few things I wanted to ask about, I probably won't ask them all. But do you look at the interactions of the dislocation loops and the precipitates?
Yeah, that's actually in that the terrible video that I showed. Mm-hmm. That's one of the things that we're trying to understand is how do those, if you have a precipitate sitting in the matrix, that precipitate has an interface, of course. That interface has an energy landscape that would absorb those defects, right? Mm-hmm. And so what we're trying to understand is how far do loops need to move in order to get there? How long does it take for that interaction to happen? All of those types of effects. So that's a project that one of my graduate students is actively working on at the moment. And what's the motivation for that, actually, that in-situ experiment? Unfortunately, she's going on internship this summer, so we won't get more data until September.
Okay, and we have a question from Jeremy. Does it take a lot of RAM capacity if you run the in-situ experiments and the YOLO algorithm simultaneously?
Yeah, I don't want to give the secret sauce for that quite yet. But what I will say is that the YOLO takes just under 4 gigs of memory run. But the way we are doing it is not running both on the same computer. So the hardware limitations is on a consideration.
Any other questions?
You can keep firing away if you have more.
I'll ask another one. So you do FIB sample prep. Does are you using I guess a plasma fib or a gallium fib, and does the damage that you create with the fib impact your experiments at all?
Yeah, great deduction question. Yeah, it totally can. So we typically will use a PFib for plasma fib for primary trenching, because it's just so much faster, right? Mm-hmm. And then we do the gallium fib for low energy final thinning in that process. If you stay for a typical FBI system, which you can start deducing that Michigan has many of these, they run at 30 kV with gallium ions. That will impart these little black dots that you see in this image, or it can. Going down to 5 kV and 2 kV, so low beam energy milling for most iron-based and nickel-based alloys, that will clean up the defects a lot and do that. And that's one of the beauties about the in-situ data is if you did have fib-induced damage, you can deconvolute that from the new damage that you're creating through the ion irradiations. But for ex-situ stuff, or to make our in-situ videos a little bit nicer, we also deploy a method called flash polishing, which is you only bring your fib sample down to about 200 nanometers, and then you electropolish it for just a fraction of a second in order to remove the last couple tens of nanometers from the sample and also remove the fib damage in there. So there's techniques to get around it. So you're totally correct. A fib is an ion beam, and so it is damaging.
Right. And so you said about 200 nanometer thick samples is what you?
If I remember right, yeah, somewhere in that kind of space. And it depends, there's a whole optimization schema you have to do for every alloy. And so some are 200, some are 120. Something you have to do pulse it at 0.1 second versus 0.12 second. There's a whole I call it black art, but there's a whole science associated with it. And even two is like for our additive manufactured samples, they will flash polish differently than conventional samples, even for the same composition. So there's a whole thing that you have to do every time you're doing this.
Cool. All right. Any other questions?
Yeah, I actually would like to ask a question on the flash polishing.
Sure.
Do you have any sources? I've looked at like PNNL is the main paper I've seen. Their M&M sort of abstract that they submitted like a year, 2 years ago.
Yeah.
If you have any publication, or if you'd be willing to forward any references you have on that, because I have some similar problems, but we haven't tried set up flash polishing where I'm at yet. So
yeah, so I completely expect that PNNL will have that a more detailed paper out in the next couple months. So that will be out there, but I'm more than happy, and I'll drop it in chat here. So if you email me after this, I will get you in contact with my flash polishing guru, who also has a bunch of recipes for different materials. And he can give you a full, I think we have presentations and standard operating procedures that I'm happy to share.
Oh, perfect. Thank you. And I have one more question. The code that is run for your in-situ analysis, is it run on the microscope itself, or is it run just sort of picking up the data as it's just being collected on a data drive?
Yeah, so everything I showed up until this last slide is all post-analysis, post-microscopy, right? So you would do the in-situ experiment, you put on a flash drive, and then you go to another computer and do that analysis. And you're using the high-end GPU systems, not like a supercomputer, but just a nice computer with a nice GPU to do that detection. This slide here is, it's doing some creative techniques in order to make it technology agnostic, right? So if you've ever played in the microscope space and trying to install software, right? Joel, FEI, Thermo Fisher, Hitachi, Zeiss, like all of them. None of them like you to install software. So we're coming up with some cute ways to get around that, but I'm not prepared to disclose it quite yet. But I have a presentation at M&M, Microscopy and Microanalysis. If you're attending that, which is in early August. If you're not, and you're in an MSA, I don't know why you're not, but you should. And we'll give some little bit more details there. Sorry, I'm being a little bit cute with it, or coy, so, but there's a reason for that.
Thank you.
Any last questions? I think we have, if there's no other questions, maybe we can come back if there are any questions after this, but I want to give Avinash a minute. He's the MSA Student Council President, and he would just like to talk, I think, for a minute about the pre-meeting Congress for M&M. So.
Thanks, Stephen. Thanks, Kevin, for that was a great talk. Thanks a lot.
Yeah.
Let me just share a few things about the M&M, as Kevin mentioned. So during the M&M, MSA Student Council also organized the pre-meeting Congress, which is especially designed for students, postdocs, and early career professionals. So this year we are going for a theme as like life as a microscopist. So we'll be starting with like sample prep, which is like critical techniques. So we'll be discussing, there will be a session for that. And what are the different cutting edge microscopy techniques, and then data analysis and how you go about publication, where Dr. John Mansfield will be talking about. And then there will be a lot of networking events. And finally, we will have some social events. So if you have not registered yet, please register. The deadline has been extended for the early registration, which is like 15th June. While you will registering for M&M, you will get an option to check the box for the PMC. So we hope that we'll get more participants this year. Thank you. Please let us know if you will have any questions. Thanks, Stephen.
Thanks, Avinash. Yeah, it is a few dollars more, I think, to register for the PMC, but hopefully your advisor will pay for that. So. Are there any last questions?
I have a question, just came in my mind about in-situ experiments, the design which you showed about the den. Is there any facility with ion radiation, with electrical measurement? I'm just asking in terms of, I work on some functional oxide materials, and people started doing radiation, and there they are seeing electrical property changes, and somewhere the properties is even improving. So is there any facility that in Michigan?
I'll put it this way, there's a possibility. So we use a den stage primarily because it works. What's the other one? It's not Hummingbird, Protochips. Protochips is the typical manufacturer you would go to for electrical biasing in-situ experiments in TEM. We have one here. We have not quite had success doing that in situ in the microscope for irradiations. in principle, it's possible. But what we think our hypothesis is why this is not working is that DENS uses an amorphous film and protochips uses a crystalline film. And the crystalline film, when it transfers from crystalline to amorphous, actually swells. That swelling percent actually fractures the chip and makes it go away. So it's a total radiation effect, like basic thing that's happening. But we haven't fully confirmed that hypothesis. So if you don't send protochips my way yelling at me at that point. But we tried about 5 chips and haven't had a success. But in theory, it's potentially possible. Just I think, and maybe I didn't press upon as much. Any of these in situ experiments, they're a little bit of luck and a little bit of science, right? So some of it is just trying to get the right amount of luck and science to happen that day to get it to work. So yeah, it's possible, but I think if there's development that would be needed.
Thanks.
All right. Well, thank you, Kevin. That was very interesting. Really appreciate you doing the talk for us.
No worries.
All right. I think with that, we'll end the meeting. Thank you very much.
Yeah.
Appreciate all you students joining us. Bye.
Bye.